Dr. Judd had a family emergency, so I don't think he's going to be here. So I'm going to take you through here at the beginning, trying to clarify some things that appeared to be unclear at the end of the class last time. So give me a sec to get myself together here.
Why are we in a circle
I
now?
was gonna enter it one time.
Oh, I hate it.
I was gonna enter points.
You carried us on good food, I'll give you that.
So what?
You carried us on good food, I will give you
I just
Oh, I barely saw you this morning. It registered, and then I couldn't think of your name. But I also haven't been able to think of going through fields all day.
I wasn't ready when we first came until like the first 3 weeks.
So I had 2 water bugs?
Are they dead?
I have one.
COVID in every class.
All right. So Dr. Judd prepared a little bit of a wrap-up of photosynthesis for you that would hopefully help you help clarify some of the things that were maybe not so clear. Why is it not showing? Hopefully clarify some of the things that maybe were a little confusing with those maps at the end. So what you need to remember is that the photosynthetic pathway that is going to be most efficient in any given situation is determined in part by how much carbon dioxide is available, and is also in part determined by the temperature that the plant is living at and what the moisture availability is in that environment. So this was in the presentation on Wednesday. As daytime growing temperature goes up and atmospheric CO2 goes down, C4 plants are favored because when carbon dioxide is limiting, most plants have to open their stomata up more often to let that CO2 into the leaf. When they do that, they lose water vapor out of their leaf, and so as it gets hotter and lower carbon dioxide levels, they're opening their stomata more. In hot environments, they dehydrate. They are not favored. When you have plenty of CO2 and relatively cool temperatures, you get C3 plants dominating because they're not then at a disadvantage when it comes to opening up their stomata, because there's plenty of CO2 to get into the leaf. And also when they do open their stomata at cooler temperatures, they're losing less water through evaporation.
So you said that when temperatures, when it's hotter and there are lower CO2 levels, C4
Low-
plants are favored?
Yep, low CO2 level and hot temperatures, C4 is favored. Low temperatures and high CO2, C3 plants are being favored.
Thank you.
So these are just general kind of walking around rules for thinking about when C3 and C4 plants will dominate in an ecosystem. Once again, as you look at total amount of carbon dioxide that is available versus the photosynthetic rate, what you see is that at low CO2 levels, you get C4 dominating. At higher temperatures, at higher CO2 levels, you get C3 plants dominating in terms of rate of photosynthesis, because the difference between this line and this line at high carbon dioxide levels is basically a measure of how much energy gets used up in the extra steps that a C4 plant has to go through to do photosynthesis. That process of fixing carbon in one cell, the mesophyll cells, fixing carbon with what enzyme?
Visco?
Not in C4 plants.
That's
PEP carboxylase. Fixing it with PEP carboxylase in the mesophyll cells, then actively transporting that malic acid out of the mesophyll cells into which cells? The bundle sheath cells. That requires energy. then stripping off that carbon dioxide so it can go into what portion of photosynthesis? The Calvin cycle. All of that takes energy, and that extra energy is the difference between the photosynthetic rate in terms of millimoles of glucose produced per unit time. That difference is the difference in the energetics of all of those extra steps that C4 plants have to take. If you are in an environment that is cool and moist, no need to evolve those mechanisms to keep your rubisco safe from oxygen, because you can just utilize the carbon dioxide that is there, open your stomata whenever you feel like, and it's no big deal, and you don't have to suffer the energetic costs of developing these methods for keeping rubisco safe from oxygen. So when we look at, this is from a different publication, when we look at the distribution of C3 and C4 plants, the reason that graphic that he showed you on Wednesday was kind of confusing, which is why he spent the last couple of days going and finding other ways of looking at this, is because there's also a seasonality aspect to all of this. At certain times of the year, even in a place where if you go to Kanza in August, you see that the place is dominated by C4 plants, but if you went to Kanza in May or April, you might see that if you look at total biomass, there may be a lot of C3 plants on the landscape. So there's also the seasonality to when temperature rises and when moisture comes. And so that can be confusing, and that's what was in a lot of that figure that he showed you. So this is somewhat of a simpler image where it basically shows the dominance of C4 plants from being totally dominant to being not dominant. And all of this would be communities with nothing but C3 plants, communities that are dominated heavily by C4 plants, and then everything in between. And what you see in this is you see C4 plants in places where you expect them to be. The Mesic grasslands of the sub-Saharan Africa. This is a region called the Sahel. It's an area where traditional agriculture has been in the form of wheat. They still grow wheat there. But this is an area that is dominated by C4 plants. You see the northern grasslands of Australia being dominated by C4. And then here in the Mesic portions of North America, you see this mixture of C3 and C4 as you move away from the moist regions of the desert southwest, or sorry, of the southeastern United States, where they're dominated by forests. You see the grasslands coming here, and these grasslands are mixed grasslands for the most part. But we showed you this figure at the beginning of the semester, and we showed you that there's tall grass prairie in the east, then mixed grass prairie, and then short grass prairie in the west. And this reflects this moisture gradient as you get further and further away from the Gulf of Mexico and the moisture influences of the Gulf of Mexico, you get shorter and shorter stature grasses. This is from another paper that kind of zooms in on this area. You see that here's Missouri here. We've got Texas down here, Oklahoma, Kansas, Nebraska, South Dakota, North Dakota. And what you notice when you look at this map is that you see this is the Flint Hills outlined here. This is an area where it is dominated by C4 grasses. It's warm enough, it's in the warm parts of this distribution of traditional prairie. It's warm enough that it favors C4 grasses. And as you move further west, you also have this band of C4 grasses over here in extreme West Texas and eastern New Mexico. But then as you move north and it gets cooler, you see it flipping from grasslands that are being dominated by C4 grasses to grasslands that are dominated by C3 grasses. And so you see C3 distribution up here more so than C4, simply because you're dealing with cooler temperatures rather than warmer temperatures. I show my ecology of food students a figure of where wheat is grown and where corn is grown. Corn is a C4 grass and wheat is a C3 grass. And you basically find wheat being grown up here along the Canadian border, and you see corn grown all throughout the bulk of the Midwest. But once again, that's a temperature relationship. Wheat grows better when you get up into southern Canada and northern United States because it's cooler, favoring C3 grasses. So when you look at Andropogon gerardii, this is, do you recall what species Andropogon gerardii is? Common name? It's big blue stem. It's big blue stem. And then Salvia pitcheri. Salvia in general, that genus is referring to what plant's common name? Do you recall? Blue sage. This is a C4 grass. This is a C3 non-grass. And people have done studies on these where they look at the rate of photosynthesis as you increase the amount of light. As you increase the amount of light, what all is happening to these plants? What's happening to their leaves? Hmm? What? Well, their leaves are always open. What's open? Okay, so when you put light on a leaf, what does the leaf start doing? Starts doing photosynthesis, so eventually you're going to deplete the carbon dioxide that's in there. You're going to need to open up your stomata to get more carbon dioxide in there. What else happens to a leaf as you expose it to sunlight?
It'll heat up.
It'll also heat up. So photon flux density is not just how many photons are hitting a leaf, but it's also an indirect measure of how much temperature that leaf is gaining over time. Andropogon gerardii is this curve right here. And as it gets more and more light, and as it gets hotter and hotter as a result of this light, it just does more and more photosynthesis. Whereas all of these other C3 plants, Salvia pitcheri, solidago, do you guys remember what solidago is? All the species of this group of organisms, this group of plants is in the genus Solidago. What's that? Goldenrods, yeah. And then I'm not sure what this other one is, the bulbini. But all of these reach some level of photon flux density, at which point they get saturated. They're at the limits of their ability to replenish CO2 in a way that doesn't basically cause them to lose too much water, whereas Andropogon just keeps getting higher and higher. So when you look at Andropogon and Salvia, which one of these is more likely to respond well to drought stress? Why? Hmm? Okay, so here it is, lowland and upland populations, burned versus unburned, lowland, upland, burned versus unburned. Is the place that you find the plant more important, or is the C3 or C4-ness of the plant more important?
It looks like the C3 versus C4-ness is
Well,
the
they're both
greater.
C4. I thought you said they were both C4. One just.
No, Salvia is C3.
I thought you said it was a C4 non-grass.
Nope, it's a C3 non-grass,
Oh.
C4 grass.
Oh, okay.
If I said that, I
So
misspoke.
basically, they're completely opposite.
Yeah, this one has really efficient water use efficiency regardless of where you find it. Kanza, this has low water use efficiency, which is what you should expect in a C3 grass. Does not use water efficiently. The number of moles of CO2 that you can create from a mole of water is very low in C3 plants rather than C4 plants. And so droughts will tend to favor C4 plants. Now, it can get too droughty, then what happens? If droughts are too severe and will go for too long, what do you turn into as an ecosystem? You turn into a desert. Guess what you don't find in deserts?
Grasses.
Grasses, period. You find other kinds of plants
Yeah.
that have different methods of doing photosynthesis that survive well in deserts. You leave the grassland and you enter the land of chem photosynthesis. What about nitrogen availability? So this is nitrogen that is found in Andropogon gerardii and Salvia pitcheri. Which of these two species do you think is going to survive better under periods of nitrogen limitation?
Gerardii.
Yeah, it needs less nitrogen. As a result, it's going to be a better competitor under low nitrogen environments. So I want you to fill in this individually and then compare your answers within your group. For water use efficiency, which is more efficient, C3 or C4? For photorespiration, which experiences higher rates of photorespiration? Which of these two, C3 or C4? What's the light intensity at which each of these things achieve the fastest, the point at which photosynthesis saturates, reaches its maximum rate? And then what is the optimal temperature for photosynthesis for each of these? Is it high or low? Somebody from BioBusters is going to give us the answers to these. Okay, that's you guys, right? Yeah.
We got a low for this one?
You,
What are you talking about, rate?
there's less of
Rate
it
or
than-
efficiency?
there's, it's,
More for
there's,
respiration, basically.
yeah, there's,
But are you talking about efficiency?
this is, rates of each of these things.
After you're done
I
talking,
said, what's the
whatever,
rate
one
of
of the three,
recipe?
one of you
That's
go.
the only thing.
That's higher.
That's the only thing that's higher.
Yeah.
But it has a higher light intensity and a higher output temperature. It's more durable.
Yeah, but
For water use efficiency, I don't think I'm understanding what the.
Water efficiency, so think of the drought mechanism.
So
Water
it's
use
using
efficiency,
water,
they use water at a more efficient rate than
Okay.
C3, so they can provide a barrel with deprivation of water.
Okay, I
'Cause-
thought about it and I was like, is it meaning that?
Yeah, like even if they have less of it, so like with C3 and C4, if they have like a little amount of water, C4 will do better because it can hold on to it longer.
the idea is that, it doesn't necessarily have to open stomata
Yeah.
until the time is right.
I get that, I just was losing the ratio in my head.
CO2.
So these are your choices of things, of answers. So which ones go in which places?
Yeah, for the respiration. Using CO2. Oh, respiration, heating CO2.
No, photorespiration is when rather than Rubisco grabbing a CO2 molecule, it grabs an oxygen molecule. And that's bad because you suddenly don't have carbon. And
Carbon, carbon.
you, no, it's that you,
There's no more CO2.
you can't
Bad
form
when
sugar.
there's no more CO2.
You can't form sugar without carbon.
You need, yeah, that's right.
So if you grab an oxygen, you don't have a carbon.
It's a useless
Yeah.
footprint.
And it plus energy.
It was a lot, and it costs the same energy. It's like the
Yeah.
same word for no. I remember, because you need the carbon, you need the
CH2O.
Yeah.
So the basic structure. So I decided to look up the V. baldwini, and it's Anastera. It's I forget how to pronounce that family name.
Asteraceae?
Yeah, Asteraceae.
Vernonia baldwini. What's coming in? Was that ironweed?
Yeah.
The Bolton one is,
Oh yeah, it is ironweed.
Did
Okay.
you say
Yeah.
low light for C4?
I recognize it to look at, so see it. I just don't know the scientific name of it.
I think that's right, cause I think it's something about like in the deserts when it like we still do photosynthesis like at night. But I don't remember what that was. But yeah, I think that was
While Gerard is putting that up, this is a good way of studying, finding ways of kind of organizing the knowledge that you're gaining, so creating charts and tables like this of how you can, especially when you have something to compare to something else, thinking about C3 and C4, and okay, well, what are the fundamental differences between them? Well, the different water use efficiency, photorespiration, light intensity at which photosynthesis saturates, and the temperatures for effective photosynthesis. And so it's easy to put this into a table and organize your knowledge a little bit rather than leaving it in disorganized forms. All right, now does everybody agree with these conclusions? Anybody disagree? Does everybody understand why these are the right answers? No, Miriam.
why is it on the third one low light and then high light?
So the light intensity at which photosynthesis saturates.
Yeah.
So this goes back to that figure that we just talked about. I know. Where as you give it more light, that's photosynthetic photon flux density is, as you give it more and more light, photosynthesis saturates, it reaches a point at which it levels off, and then giving it more light doesn't improve things because it's already saturated. It's doing the maximum amount of photosynthesis that it can do given the amount of rubisco and the amount of carbon dioxide that it can let into its leaves. Whereas Andropogon gerardiae continues to go up because as you're adding light, what are you also doing to the plant? You're making the plant, Elizabeth?
Hotter.
You're making the plant hotter, so as these plants get heated up, they don't open their stomata as much because they are risking dehydration as water vapor escapes from them, and so they don't pull in as much carbon dioxide. These guys can continue to pull in carbon dioxide because they use the full amount of carbon dioxide that are available in the air spaces in the leaf before they open their stomata up. As they deplete carbon dioxide levels, you go down to practically no carbon dioxide within the leaf, and even though carbon dioxide is at relatively low concentrations in the atmosphere, the more you deplete carbon dioxide in your leaf before you open your stomata, you make that concentration gradient steeper. So what happens to the rate of diffusion when you have a steep concentration gradient rather than a shallow concentration gradient? How is diffusion rate related to the difference in concentrations in the two areas? Basic chemistry, yeah. What?
Steep rate happens quicker.
Yeah, if you have a steep gradient, diffusion rate happens quicker. So if you have a 10% salt solution here, and you have a 90% salt solution here, you're going to see, and there's a semi-permeable membrane through here. Maybe you have, we used to do these experiments in freshman biology that were stupid now that I think back to them, but they got these points across pretty well. If you can get dialysis tubing, moisten it, and then put 2 concentrations here on either side of this, this is gonna be much faster diffusion across that membrane than if you have a 48% salt solution and a 50% salt solution. The net movement of salt is still gonna be from here to there, but for every molecule of salt that goes one direction, you're going to have a lot of molecules of salt going back the other direction, so the net rate of diffusion is going to be lower. For every molecule of salt that goes this direction, there are very few salt molecules that are going that direction, so the net rate of diffusion is faster. So when you deplete the leaf of all the CO2, when you do open the stomata up, CO2 just goes flooding into that leaf, so you can open your stomata less often because you don't have to open it up because you don't have oxygen competing for carbon dioxide, which means that you can completely deplete your leaf of carbon dioxide. So you open your stomata less often, but then when you do go to open your stomata, you open them for a shorter period of time because this concentration gradient is steeper. And so for all those reasons, you get saturation occurring at higher light levels than lower light levels. Anyone else need to understand why these are the right answers? Yeah, so optimal temperature for photosynthesis, it's related to what we just did. As those C4 plants got hotter and hotter, they could continue doing photosynthesis because they don't risk losing as much water vapor from their leaves because they open their stomata less often, and when they do open them for a shorter period of time, as opposed to C3. So C3 are going to have lower temperatures where photosynthesis is optimal, and C4 plants can sustain photosynthesis at higher temperatures. So that figure that we just talked about, it's not just how much light is hitting the leaf, but it's also how much temperature that leaf is sustaining because of more light. Does that make sense?
Yes.
Okay. All right. So we have talked about C3 plants, where all of the mesophyll cells do photosynthesis the same way, are involved in carbon fixation in the same way. We have C4 plants, where carbon fixation is segregated out in these mesophyll cells, and then once carbon is captured, you then transfer that carbon capture into the bundle sheath cells, and the bundle sheath cells is where the Calvin cycle goes on. This is a spatial segregation of these processes. When we talk about the light reactions, when Dr. Judd was talking about light-dependent and light-independent reactions, what was he drawing a distinction between? What are the light-dependent reactions? What's involved in that?
That is turning, that's using the light energy to break up height, to excite electrons with, and then using water.
Yeah, so you get electrons from water.
Yeah.
You use the light to basically capture the electron, get the energy from the light that is held in these excited electrons to create what compounds?
Oxygen.
Well, oxygen is a final electron acceptor, but what energy containing compounds are created in this
ATP?
process? ATP and NADPH. And then what is that ATP and NADPH used for?
To churn the Calvin cycle.
To turn the Calvin cycle, yep. So all that energy is then used for the chemical reactions that are involved in the Calvin cycle. We used to call these the light reactions and the dark reactions, but we now call them the light-dependent reactions and the light-independent reactions, and it's because the generation of ATP and NADPH cannot go on unless you have light, which is why they're called the light-dependent reactions. The light-dependent reactions, which is initial carbon capture and the Calvin cycle, that can occur whether there's light or not, as long as there's stored energy. Dr. Judd told me earlier today, he kind of views the light reactions as charging the battery, and then the energy that is in the battery gets used for the carbon capture and the Calvin cycle. So CAM plants, which we haven't really talked about, but these are things like cacti and the Euphorbiaceae in the old world, they have CAM photosynthesis, and rather than there being a spatial separation between initial carbon capture and the Calvin cycle, they separate this process in terms of what time of day the plant does this. And carbon capture goes on at night because carbon capture is a light independent sort of reaction, and the Calvin cycle goes on during the day when the energy from the light dependent reactions is available. So it does initial carbon capture, uses PEP carboxylase rather than Rubisco during the night to capture carbon, because it can open its stomata in a desert at night when it's cool and moist, and it can open its stomata however long it needs to get as much carbon dioxide into the leaf of the plant, the stem of the plant, so that it doesn't have to worry about losing water vapor during that time. So initial carbon capture is only captured using PEP carboxylase, so it doesn't get interfered with oxygen. And then during the daytime, it just shuts all of its stomata and then takes the carbon dioxide from malic acid and then gives it to Rubisco, and then depletes all of the carbon dioxide that is released from malic acid in the process of doing the Calvin cycle. Yeah.
So the CAM plants are specified their cycles in night and day.
Yep.
But the C4 plants aren't necessarily night and day.
Nope.
They're not clear opposites.
They're just so one way of thinking of it is in C4 plants there's actually a spatial segregation, mesophyll versus bundle sheath cells.
There's no
Here it's all
bundle sheath,
happening
no
in
bundle
the
sheath.
same cells, but there's this temporal separation. You do initial capture, carbon capture in the nighttime when you can get access to more and more CO2 without losing a lot of water vapor because you're not hot, and so it's the coolest, moistest time.
And that's why it does it. It's the vacuoles, not the bundle sheath. The
Sorry,
CAM
what?
The CAM, all that occurs in the vacuoles.
But this all occurs in mesophyll cells.
This isn't.
Where do you see vacuoles?
Yeah, like those vacuoles can concentration carbons in vacuoles.
Oh, in vacuoles, yes. Yeah, in the central vacuole inside the plant
Those
cell.
are in vacuoles, yeah.
Yeah, so if you look at the structure of a plant cell, the cytoplasm is all squished to the side. There will be a nucleus. The chloroplasts circulate around in the cytoplasm, but inside the plant there's this large central vacuole that animal cells don't have. Yeah, and that's where it concentrates its CO2. So that finishes up photosynthesis. Any questions about that? Because you lose water vapor through stomata, plants have varying degrees of stomatal density on their leaves. And so you might expect to have stomatal density changing over time in response to what kinds of physical parameters might affect stomatal density. Go ahead, James.
CO2 concentrations?
So if CO2 concentrations go up, what might you expect to happen with stomatal density? You might expect it to go down because if there's more concentration of CO2 in the environment, you don't need as many stomata to get CO2 into the plant. And if you reduce the number of stomata that you have, you reduce overall water loss. If warming is occurring, what might you expect to happen to stomatal density?
Increases.
you might expect it to decrease. Once again, it's a water saving sort of issue. So Dr. Judd wanted me to tell you about this citizen science opportunity. They have photos of ginkgo leaves taken from the fossil record, and you can just log on, get some photos, and count stomata on leaves that are taken from different parts of the fossil record, because they're trying to correlate stomatal density with what we know of temperature from things like ice cores. And because ginkgos are a relatively ancient plant group, you can actually then go to time periods in evolutionary history before we have ice core data, and then use stomatal density to then project what even longer term temperature dynamics were. So anyway, feel like playing around on a citizen science project that has to do with this very issue. There's something there available for you. The link is in the PDF that's posted. All right. So next phase, for some reason Dr. Judd got away from doing the plants and animals, so I'm going to start that back up. Prairie grass of the day. This is Panicum virgatum switchgrass. Switchgrass has become something of a fad here in the Midwest because people are growing switchgrass to hopefully generate biofuel out of it. Switchgrass can grow really fast. It's a C4 grass. Grows really fast. It has this really characteristic seed head inflorescence at the top of it. It's well known for having deep roots, which is also true of big bluestem and little bluestem and a lot of our plant species here in the Midwest. Deep root or perennial rather. We've already talked about what it means to be rhizomatous in terms of how it can respond to disturbances like fire. Some people are growing switchgrass as a potential biofuel. Some people are growing switchgrass as forage. There's a program at University of Missouri-Rolla. I guess it's not called that. I guess it's Missouri State Science and Technology University. Anyway, they have a couple of labs there that are exploring switchgrass as a source of biofuels. I chose a forb of the day that happens to be a legume. So remember, legumes are in the family Fabaceae. This is purple clover. It's common in prairies. It's hard to see the structure of the leaves that kind of are indicative of it being a legume, but it's a perennial herb. It will grow back year after year. Has a very large taproot. It was used by Native Americans for food. They would pull up the taproot and eat the taproot. It's used in restoration of lands because it is a nitrogen-fixing plant, because it is a legume. It has these nodules that have nitrogen-fixing bacteria associated with them, and so you can grow prairie clover in an old field and restore nitrogen availability to the soil relatively easily. And it's adapted to this environment, so it grows better than some other clovers might, some other legumes might grow. The prairie animal for the day is a bull snake, Pituophis melanoleucus. It's called melanoleucus, the scientific name, because it's darker at the front end than it is at the back end. It's a very common snake that you see in prairies in the Midwest. It has a color pattern that makes people mistake it sometimes for a rattlesnake, and so they will mistakenly kill bull snakes. Also, it makes a hissing sound that can also be acoustic mimicry. It's thought to be acoustic mimicry of rattlesnakes, which might scare away a non-human animal, but all it causes a human animal to do is to beat it with a shovel.
They're non-venomous though, right?
They're non-venomous. They're perfectly harmless. This is how perfectly harmless they are. Somebody who has never touched a snake in their life before is touching a Pituophis melanoleucus at the National Tallgrass Prairie Reserve down in Kansas. They feed on small mammals. They especially like pocket gophers. They can lay quite a few eggs late in the year, and they can get fairly long, but even when they're fairly long, they're not a terribly beefy snake. If you had a rattlesnake that was 6 feet long, that rattlesnake would be really, really thick. These are not going to be terribly thick anytime.
So it's more like a gopher snake?
It is a gopher snake.
Oh, okay.
That's another common name. This is why we don't use common names.
Let's say it looks just like a gopher snake.
It does look
It's
just
more
like
like
a-
a gopher snake than a rattlesnake, but I guess.
Yeah, Louisiana pine snake is an endangered subspecies that lives in the eastern woods of Texas. Yeah, gopher snake, pine snake, all these are names for the same organism. So, prairie plants and animals of the day. It seems like it's been a long time since we started talking about Kanza. It hasn't been that long, but it might seem like a long time. So before we go into nutrient cycling, I want to kind of remind you of why we're covering the kinds of topics that we're covering in the Kanza Prairie case study. So what we're talking a lot about in Kanza is the plant community. When you were asked to calculate species diversity indices, all of those species diversity indices that you calculated, those were species diversity indices of the plant community, not the animal community. So when we think about the plant community or the plant and animal community, we oftentimes think of plants in terms of above ground biomass. Now this above ground biomass might be made up of lots of different species. It could in theory be made up of a single species, but included in above ground biomass is all the biomass of all the C4 plants, all the C3 plants, all of the legumes, all of the nitrogen fixing legumes, all of the non-nitrogen fixing forbs that are in the prairie. But ultimately, the amount of above ground biomass that you have and the species composition that makes up that above ground biomass is going to be influenced by external drivers and internal drivers. Drivers that are external to the system and drivers that are internal to the system. The big driver that is external to the system is climate. A lot of times we boil climate down to temperature and rainfall. And as we talk about C3 versus C4 plants and how they dominate in ecosystems, it's really easy to see how temperature and rainfall affect not only how much above ground biomass there is, but also who makes up that above ground biomass. Is it C3 plants? Is it C4 plants? This above ground biomass can be reduced, at Kanza at least, through two types of disturbance, fire and grazing. But these two kinds of disturbance have different effects on above ground biomass. They both reduce above ground biomass in that they decrease biomass, but grazing decreases biomass selectively in that bison prefer things like warm season grasses, things like big bluestem and little bluestem. Fire, though, decreases biomass and is totally unselective. Everything burns up when a fire goes through a landscape. So grazing results in selective decreases in biomass. Fire results in unselective decreases in biomass. The reduction of biomass, though, in both of these cases alters the light environment. After a fire and after grazing, what happens to light availability? It goes up. If it goes up, what happens to competition for light?
Goes down.
It goes down, or it's different than it was before. Some species will thrive more in a high light environment than other species will, for all the reasons that we just discussed there at the end of photosynthesis. That altered light environment changes competition for light. It then affects how much biomass is there and who makes up that biomass. Luke, you have a question?
It says under here, grazing and fire both decrease biomass.
They do.
Grazing is selected because the bison are choosing what to eat.
Yes.
Fire is unselected because it just burns.
Just burns everything. Yep.
Yeah.
Yeah, so bison tend to eat warm season grasses, which then creates space for forbs to grow in, which is why you tend to have more diversity in.
Because it's eating a common plant.
It's eating a common plant, yeah.
And
And then it frees up some space, some competitive
gives
space,
rise for some non-dominant plants to
Yep.
come in and grow.
which is why when you did your exercise on plant diversity and you compared bison grazed areas to non-bison grazed areas, bison grazed areas have more species diversity because it removed those dominant plants and as a result made space for the less dominant plants to grow. So it changes the light environment, which opens up ecological space that then affects who is around making up this above ground biomass. Grazing and fire will also both alter nutrient availability, but they're going to alter nutrient availability in different ways. When these grazers are here on the landscape, how do they alter nutrient availability? Yeah, right.
They can increase
they're pooping and peeing on the landscape. Feces, stool, whatever, scat, cow pats, whatever you want to call it, those are generally high in phosphorus, and urine is high in urea, which is a nitrogen-containing compound. So the two compounds that are most limiting in a pasture, in a prairie, which are nitrogen and phosphorus, are both excreted by large herbivores.
Isn't ammonia present in urine as well?
Yep, ammonia is also, but organisms can absorb all of that. Yeah.
Animals can also like transfer seeds from like one area to the
Animals
other.
can also transfer seeds from one area to another. That's another potential aspect of grazing. Although in prairies, I think it's probably a lot more hitchhiking. Things like tick trefoil that sticks to the fur. I think there's a lot more of that kind of transport than them eating seeds and then pooping them out someplace else. It's not that it doesn't happen, it's just in this system, I think that happens more. If you go to, for example, the Guanacaste forests of western Costa Rica, a lot of that seed dispersal is actually organisms eating fruits, eating the seeds while they're eating the fruit and then going and pooping that out someplace else. A famous guy named Daniel Jansen used horses to simulate a loss of native frugivores that happened to kind of perpetuate the Guanacaste forest by bringing horses in, letting them eat the fruit, they walk around, they distribute the seeds. It turns out the seeds don't really germinate very well until they pass through the gut of an animal. Similar thing happened on the island of Mauritius. On Mauritius, they have these gigantic birds called elephant birds. They were hunted to extinction by sailors that came and stopped off in Mauritius. They were big birds, they were dangerous, they would shoot them, kill them, bring them onto the boat, and eat them. And when they went extinct, 100 years later, they discovered that the trees on the island were old, and as they were dying out, there weren't new seedlings coming in to replace them. And there was a biologist who had an idea that maybe the reason that the forest wasn't regenerating as the forest trees died off was because the moa had been driven to extinction. When the moa was driven to extinction, there was nobody to eat the fruits. The seeds didn't go through the gut of the moa. They didn't get the seed coat weakened as a result of digestion, and as a result, they were not germinating. And so he basically took some seeds from the trees, dunked them in beakers full of acid for a period of time, and then planted them, and they started germinating. And so sometimes these herbivores can be really important. I think that's less of a thing on Kanza than it is in other places. Yeah.
You said that grazing increases nutrient availability by their dispersal of their urine and their
Yep.
defecation, but it also can decrease it, right, in some spots?
It can
Like
decrease
near,
it.
like near water sources and near shade, things like that.
Well, bison really don't like shade.
You know?
That's a difference, a fundamental difference between bison and cattle. Cattle, when it's hot out, they will preferentially go down around bodies of water down in creek bottoms, and they kind of tear up creek bottoms and lead to a lot of erosion and things like this. If you ever go out to Kanza or go to a ranch that raises bison, in the heat of the day in the middle of August, you will see bison at the tops of the hills. They don't really go down and hang out around water sources like domestic cattle do.
So they're less destructive to land.
They are super less destructive to land.
You know?
And so,
There's patches like with cattle that they're just horrible, because that's where they hang out all the time,
Yep.
their weight. They
yeah, the only place where bison really disturb the soil at all, that might influence nutrient cycling, is in these places where they wallow. So bison wallows are fundamentally different than the rest of Kanza in terms of depth of soil and nutrient content of that soil. The other way in which bison contribute to nutrient cycling is when they die.
Yeah.
When they die, that's a huge pulse of nitrogen and phosphorus to the soil in that area right around it. To the point that where they die, they kill whatever plants are underneath them, because those plants are now shaded. But even for a time period after that, there's too many nutrients in the soil for plants to come back. And so you'll see these concentric rings of plant growth radiating out from the body, the carcass of the bison, as the nutrient content of the soil attenuates. It's a pretty cool thing to see. So yeah, we would normally think of grazing as increasing nutrient availability through excretion of metabolic waste. Fire, once again, has a dual sort of influence on nutrient cycling, in that when you burn up a bunch of plant biomass, a lot of that biomass is volatilized, it's sent into the atmosphere, but the ash that is left behind still has nutrients in it, but because it's undergone the process of burning, those nutrients are much more available than they are if you were just gonna wait for that stuff to decompose. So fire has the dual process of both allowing nutrients to escape from the ecosystem through volatilization, but also making nutrients available faster because ash decomposes faster than non-ashed
And another
material.
thing too with like fire, isn't it that combustion action is kind of like just combusting those nutrients throughout too in the ash?
No, it either volatilizes them or it converts them into ash that then gets absorbed pretty quickly by the soil. But I'm not sure what you mean by combusting
I
the
thought
ground.
combustion was, the process of combustion was promoting growth. By
To promote-
spreading nutrients everywhere.
No, it doesn't spread nutrients everywhere. It allows some nutrients to be more readily absorbed into the soil because it's been converted to ash, and that.
But you have to have combustion, well, I guess it's what fire is, but the ash is a product of combustion.
Ash is the product of
Yeah.
combustion, yes. So when you think about,
So the more fire you have, the more ash, the more growth.
yes, so that those nutrients are immediately available because fire is a much faster process than decomposition.
Yeah.
I mean, when you think about it from a carbon standpoint, Dr. Judd gave you these equations. No, that's all. Never mind. What's this the equation for?
Combustion.
Glucose combustion.
It's the equation for combustion. What else is it the equation for?
Respiration.
Yeah, respiration. Using glucose to produce.
So when you think about a plant, think back to the structure of a plant. What's in the cell walls of plants?
Cellulose.
Cellulose. What is cellulose?
Sugar.
What
Starch.
sugar?
Oh, wait, no. Glucose.
It's glucose. It's glucose that's bonded together in branched chains,
Yeah.
and the chains that are branched in a way that we can't digest it. We can't break down cellulose. When you eat broccoli, because you want to have a nice healthy colon, the reason you eat broccoli to have a nice healthy colon is that there's a bunch of the biomass of broccoli that you're incapable of digesting because it's made up of cellulose. 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 You're going to eat all the wood you want, and you're not going to break it down because organisms that eat cellulose have special bacteria that have enzymes that allow the bacteria to break down cellulose. It then releases the glucose, and then that glucose is available to the cow. When you eat cellulose, you don't have those bacteria, so that glucose isn't available to you.
Is
Yes.
sap cellulose?
No, sap is not cellulose. Wood is cellulose.
What is it?
Sap is sugar.
Yeah.
Wood is cellulose.
What was sap?
Sap is plant fluid going up and down the tree, and it's basically sugar dissolved in water, essentially. When you eat maple syrup, you're basically eating concentrated tree sap.
Yeah.
So in the early spring, when you go to tap maple trees, you drive a tap in there. What you're doing is you're driving the tap into the vascular system that takes energy from the roots and transfers it to the leaves.
Pocket liquid.
And you're tapping into that supply. As the sap rises, it flows out. They collect it in these big barrels. Takes, I think, 40 gallons of tree sap.
How deep do you have to go into the tree to?
Yeah, it's right there at the surface.
Uh-huh.
It's the vascular tissues which are right at the edge of the tree, right underneath the bark. And so you get 40 gallons of tree sap, essentially, that you then boil down over a period of time to about 1 gallon of maple syrup. So sap is just tree water that has a bunch of sugars in it because sugar is the way that the tree stores energy. But this tree also stores energy in the form of glucose because glucose is the cellular component, is the molecular component of cellulose, and cellulose is the structural component of the tree, and it's wrapped around every cell. So wrapped around this cell is a bunch bunch of, for most organisms, undigestible cellulose. And so trapped within this undigestible cellulose is all the nitrogen and all the phosphorus of those cells. When you burn something, you're burning the cellulose away as well as the contents inside. So that makes the nitrogen and phosphorus that's inside that cell much easier to access. Whenever we talk about the availability of nutrients to organisms, we refer to the carbon to nitrogen ratio sometimes. And this is sometimes like 500 milligrams of carbon to 1 milligram of nitrogen. That represents that there's a lot of cellulose that you have to get through to get access to that little bit amount of nitrogen. If you have a plant that has a 250 to 1 ratio of carbon to nitrogen, it's easier to access that nitrogen because there's less cellulose that you have to go through to get to that nitrogen. So carbon to nitrogen ratios influence how likely you are to be grazed on by a grazer. Grazers will tend to prefer plants that have lower carbon to nitrogen ratios than ones that have higher carbon to nitrogen ratios. And also decomposition occurs faster in low carbon to nitrogen ratio plants. Yeah.
How do they know which one has the ratio?
Oh, it's really pretty easy. We put it in a furnace and we burn it and we get the ash. And you can look at the ash, and the ash that stays behind is usually the nitrogen and phosphorus containing parts of the plant, and the cellulose containing parts go away. And so people who study this will actually do nutrient analyses on these things. And because most of the carbon that is found in a plant is bound up in cellulose, that's a good measure of the carbon that's there.
But didn't you say some would get volatilized?
Well, it doesn't get volatilized when you do this in an oven in a lab. You
So it's
do it in a,
you don't give it any room to escape?
you don't give it any room to escape.
It's like
Yeah,
chambers,
you'll
just like chambers of burning.
Yeah, yeah, so you'll see, so what happens is,
Like a barometer kind of thing?
no, it's not. It's a chemical, we actually have this capacity, I think, down in the chemistry department. What you do is you grind up, you dry mass to constant mass.
Like GC analysis?
Yep.
Okay.
You
yeah,
dry it up, you put it into a GC,
Yeah,
and what does that do? It burns up the stuff that's in there.
but it puts a pressure into it.
But it also burns it as well.
It does? I didn't know that.
Yeah.
Wow.
And what it does is it analyzes the gases that come off of that.
The pressure of the
essentially what
gas.
you're measuring.
The pressure of the gas is the reading.
Yeah.
Yes,
Yeah.
okay.
So it burns up your sample. That's why you can't
That
ever run.
combustion pressure, that'll tell you what it is.
Yep. It tells you what the compounds are.
Because they each
and it
have a
tells you-
certain, they each have a certain pressure.
Yep.
Holy crap.
So you can do these analyses and see what has a high carbon nitrogen ratio versus low carbon nitrogen ratio. So fire is complicated. It both makes nutrients available and it also causes nutrients to be lost from an ecosystem because of this volatilization. Man, that took us a long time just to get through the overview. So when we were talking about all of the topics that we've talked about so far in this class, we've talked about climate at the beginning when we introduced Konza Prairie to you. Dr. Judd has been talking to you a lot about photosynthesis because that's what builds above ground biomass. I talked to you for a while about disturbance because these are the two kinds of disturbances that we have. And then I'm going to be talking to you on Monday about nutrient cycling because both of these disturbances affect nutrient cycling in different ways. So what I guess I want you to do for homework, this is what we're going to be doing. We're going to be talking about pools and fluxes of these different nutrient cycles. The carbon cycle is the easiest cycle to learn from because pools are places where you find a particular nutrient in an ecosystem, and pools of carbon are relatively easy to figure out. What's the pool of carbon in plants going to be like? Well, it's going to be a lot of cellulose. What is the pool in animals going to be like? Well, it's going to be a lot of glucose, but it's also going to be like lipids and proteins and things like that as well. Fluxes are movements of nutrients from one pool to another. The soil is a pool. And a transformation is a flux, a movement of a material from one pool to another pool, and in the process of that movement, it also gets transformed. So when a plant takes carbon dioxide from the atmosphere and turns it into glucose, that is not only a flux, the carbon is moving from the atmosphere to the plant, but in the process of that movement from the atmosphere to the plant, it transforms from carbon dioxide to glucose. When an animal eats that glucose, metabolizes that glucose, and breathes out carbon dioxide, you're once again taking a flux, taking carbon that is in the animal and converting it to carbon dioxide that is in the atmosphere, but it is also a transformation because it's going from glucose to carbon dioxide. But the cool thing about this is that these are the two fluxes, cellular respiration and photosynthesis. Those are the two transformations. So carbon is relatively easy to follow. So what I want you to do, without looking at the carbon cycle that is in the PowerPoint presentation that is on Moodle, I want you to take a first cut at a carbon cycle. So keep in mind that there will be organisms that live on the surface of the soil, there will be organisms that live in the soil, and then there are things like water runoff and things like that that can take nutrients out of the soil. And then up here also is the atmosphere. So take your first cut at the carbon cycle. Carbon dioxide in the atmosphere, forms of carbon in organisms, how that carbon gets into the soil, where does that carbon go once it's in the soil, et cetera, et cetera. And we'll start there as kind of a jumping off point, because I have a really detailed carbon cycle for you, but it might be useful to try and figure this out on your own. There's a reading in the daily schedule that you should read that basically also runs you through these things. So use your textbook to a certain degree, and we'll begin there on Monday.
So, Dr. Schwartz.
Yeah.
Why do we only measure above ground biomass? Why don't we include underground biomass?
We could. Above ground biomass is way higher than below ground biomass, because above ground biomass is going to be,
Are those our?
yeah, I don't know whose this is.
It might be mine. I don't
There's
think
no
I put
name
my-
on it.
I don't think I put my name on it.
Is that yours? Bronson
Yeah,
is
fine.
not here. Jonas, Lucy, Riley. Oops, sorry. Luke. Carly is not here. So below ground biomass is really hard to measure because it's mainly microbial. So,
That was off page 20.
Miriam,
We're ready to please community.
Jackson, Noah.
Because I wrote down all of the, like, this one I got
Oh, it
very
wasn't on
wrong.
the staff.
But this one I was
Yeah,
totally
It's
wrong.
something,
Listen off the
I told them.
Where?
Check with him when he gets back.
I should have.
So Jonas, it's really hard to measure because it's mostly microbial,
Okay.
and it's probably really way huger than we think it is. Because every time we think about microbial systems right now, we figure out as soon as we know a little bit, what we thought we knew is so
I
much
have
off the
two
mark.
performances tomorrow, but I'm just gonna sleep until I have to
Below
be
ground
there.
biomass is probably way huger than we think. It's just been a black box.
So it's just, even if we wanted to.
If
be too
Archer doesn't
crazy to try and measure that.
Well, it's not crazy to
That's
try
really
anything.
fair.
It's just hard.
Yeah.
So I have a former student who works at the
I,
University of
it is
Montana
opening
now,
night for me.
I'm at.
and he studies microbial systems in soil.
Valid.
Okay.
And
I ain't erased mine.
It's just that.
even though he